Welcome to Lecture 5 of Biology 121, Human Biology and Evolution. Today we're going to talk about human variation, specifically evaluating the question of whether or not human race is a genetic concept. I'm going to give away the ending, and then throughout lecture I will reveal the details that lead to these conclusions. That biological race is not a scientifically valuable way to describe human variation, and therefore race is not a genetic concept. What is race? How do biologists use this term? Well, we kind of don't anymore. It's an old-timey biology word that used to describe what we now call subspecies. And these are geographically isolated groups or populations that are headed towards speciation, but they haven't fully speciated yet. Here's an example. You can see these winter wrens. It's a type of bird. And 3 different groups exist in North America. You can see 2 of the group's ranges here on the map, and you'll notice there's an eastern range and a western range that is separated in the middle where none live because they cannot live in the Rocky Mountains. So we have an eastern winter wren, a western winter wren, and then there's one in the Aleutian Islands north, that's near Alaska. And none of these subgroups interbreed simply because they are geographically isolated. So they used to be one group, and then due to historical climate change, they became separate, and now they're evolving independently of each other in these 3 lineages. So we call them subspecies, or you might call them races. As a review from last lecture, which type of speciation produces races? Sympatric speciation or allopatric? Ta-da! Allopatric. I hope you said this. This is when geographic isolation prevents interbreeding, and over time that means each lineage is accumulating separate sets of mutations, kind of by random chance, and eventually they might become separate species. So if we're talking about humans, there are some historical separations among human populations due to impassable deserts, huge mountain ranges, and oceans that kept ancestral populations isolated from one another. There was no migration across the Himalayan mountains, for example, or across the Sahara Desert, or across giant oceans. And these geographic barriers reduced gene flow so much that ancient human populations evolved separately in different big geographic areas. Our question today asks if this separation lasted long enough to produce biologically distinct human groups in each of these regions, and therefore do genetic subspecies exist? Recall the historical biological definition of race. Here is a quote from famous biologist Ernst Mayr, who called races phenotypically similar populations of a species inhabiting a geographic subdivision of the range of that species and differing phenotypically from other populations of that species. The wrens were a clear example of this, but do these groups exist among humans as well? Could races have evolved when groups were isolated in different locations? Perhaps on different continents during ancient human history? From this historical perspective, there could be deep divisions among human groups. And historically, we might call these types of humans that are located in separate geographic areas on the maps that have historically been separated by mountain ranges, deserts, and oceans. And so here I've circled on the map some historically isolated patches of Earth where lineages could have evolved unique genetic profiles within each location. And so these circles on the map correspond to the solid colored circles on this Venn diagram representing types, in quotation marks, of humans from different continents. And then these types of humans based on genetic separation historically have only recently been blurring the lines through recent mixing, and what that refers to is interracial relationships. And so here's a famous interracial couple. Do you recognize them? Your hint is that this photo was taken in Hawaii. Here is the mom with their son. Do you recognize them now? And here's the son all grown up. This historical perspective, I hope, reminds you of a previous lecture in which we talked about this Platonic theory of forms that suggested all organisms have a celestial prototype from which all variation represents an imperfect reflection. So human types, this idea that there could be types and that each type is different from others, is really a Platonic perspective. And that contrasts our Darwinian perspective, which we'll talk about next. The Platonic perspective and the Darwinian perspective represent two scientific hypotheses that we could test with genetic evidence. Here is a different perspective. Remember that a Darwinian perspective would say types are an abstraction and that variation blends from place to place. Well, from this Darwinian perspective, we would expect a branching pattern of evolution like a family tree that has one common ancestor of all humans. We might call that the root node of a phylogeny. After that, there would be some splitting of ancestral populations into branches representing separate lineages. And these branches would be geographically isolated so that there's no gene flow, meaning no migration and no interbreeding between groups isolated in different locations. Then within each branch or each lineage, there would be random mutation and then selection, both negative selection to weed out deleterious or dangerous mutations, plus positive selection to maintain and increase the frequency of mutations that are beneficial in that environment. And in this way, adaptation would occur over many generations within each branch. And then only later, when modern world travel came about, would there be contact between these previously separated groups of people, gene flow, and intermarriages. So what would this phylogeny look like? And I hope that you understand phylogenies from the previous lab activity, so you could sketch what this would look like. And here's what it would look like. There'd be a common ancestor of all humans sometime after the split of humans from chimpanzees, and then time would pass. And in this time, this group of very, very early human ancestors would accumulate genetic variations that are specific to humans that separate us from chimpanzees. And all human groups alive today would share this human variation. And then once you get to the second node, there would be some branching. So branching of one small ancestral population into multiple groups, followed by isolation of these groups. So they split and migrated into different regions, and then they were kept separate. Over time, independent evolution could have occurred within each lineage, and these lineages may have evolved into separate subspecies, which biologists could call races. And here, these blue stars represent some set of mutations that is specific to the individual branches. And then I have this scale on the top representing biological similarity. And that just means that if the branches are short, everybody in that population is really similar. But as the branch lengths get longer, they have more and more mutations within that lineage. So over time in this Darwinian model, due to genetic drift and new mutations popping up and natural selection, this could eventually lead these lineages to diverge so much that we would call them different races. And this would mean that people were becoming more and more similar within a race and more different between races, because remember, each isolated lineage is accumulating its own sets of race-specific mutations. And that means that as people become more and more dissimilar between groups, they're losing the shared variation that they all inherited from their common ancestor. So if you were looking at their genetic sequences like you did in the lab with the 6 different animals, and you were comparing those letters down the column, more and more mutations would make the columns look less and less alike over time. And then over time, within each lineage, populations could form. And so within each race, you might have multiple populations, and these could be like different ethnicities within a continent. It's just a population is a smaller subgroup of people that tend to live near each other within a larger geographic space. So it'd be like a city within a country. But would we say that populations also constitute their own race? Like, where do we draw the line? Remember, Darwin suggests that types are a construct, an abstraction, and so the scientist has to decide where we draw the line for both species membership and membership in biological subspecies, which we could call races. So where do we draw that line? Okay, so that's the theory. If we jump ahead 100 years to the 1970s, this guy, Richard Lewontin, and others were inventing new techniques to study genetics for the first time. And he is one of the first people who tested the idea of independent evolution within isolated human groups. So he was testing this Darwinian model of race to see if it was true or not. Lewontin predicted that if people have been separated for a long time, then each race will be genetically different from every other race. Each population within a race will be genetically different from every other population in that race. And under this model, if a long time has passed, then there'd be lots of accumulated genetic differences. So lots of blue stars separating both the 4 branches that represent races and all the little branches over on the right that separate populations within the larger racial groupings. So he did some statistics called an analysis of genetic variance. And he predicted there'd be a small percent of variation among all humans that is shared. There'd be a large percent of genetic variation shared within a race, but that differs from one race to another. And he'd also expected some large differences between populations within each race. And so if we look at the genotypes, the genetic sequences of a bunch of people from all these different populations alive today, we can group them based on if most of the letters in the DNA are the same within populations, but different from other populations, or all the same within races, but different from other races. Or perhaps if we look at all of these different populations, they have some letters. If we went down the column for all the different sequences, they'd be identical. And so Lewontin predicted under this Darwinian model of races existing that there'd be few pieces of DNA left that were identical for all the people he studied today in all of these different populations. But that's not what he found when he looked at people's blood work. It turned out that most of the A's, T's, C's, and G's of people's DNA are identical. If you take any people from anywhere around the world and you compare them, their genetic sequences, 85+% of those letters are going to be exactly the same. And only 6% varied predictably by race, and within race only 8% varied between populations. So let's X out his model, and what he found instead was evidence that so little time had passed since human groups moved into these geographically isolated regions that we haven't had enough time as a species to accumulate big differences in our DNA. So we think that little time has passed since we were all one big gene pool. And that's because most of the variation seen is shared by all humans. It's what makes us human. It's identical among all of us, but it's different from chimps. To formalize his interpretation, I'll say it again. It suggests that humanity is too young to have evolved big differences between groups because mutations accumulate so slowly. For example, you could think about it this way. If a malevolent alien wiped out all human populations but one, that one population would still harbor 85.4% of the genetic variation of our entire species. So Lewontin, in his famous scientific article, said that racial classification is now seen to be of virtually no genetic significance. His results rejected a biological concept of human race. Lewontin assumed that human genetic variation looks something like this tree, but what does it look like really based on modern genetic analysis where we look at the entire genome of people from different populations? That information wasn't available to Lewontin, but it is available to us since about the year 2001, when the Human Genome Project was complete. In the early 2000s, a group of scientists visited all of the populations seen with the colorful dots on the map here. Now, these populations are special because they are meant to be indigenous groups. That means they are people who have lived in these geographic regions for many, many generations, and the oral histories of these groups of people suggest that they didn't migrate recently. So it's important to take genetic samples from people who can trace their ancestry to the region where they currently live. And then these genetic samples were run through a computer system that compared all of their A's, T's, C's, and G's, and the computer, not a human, popped out a family tree. And this family tree is what we think human genetic variation really looks like. Are you excited to see it? Drum roll, please. It's like this. Ta-da! So let me explain the tree that you're looking at. There is a long root that suggests there was a small ancestral population about 2 million years ago where all humans were intermingling in one gene pool. And the branching didn't start until much later, only 150 to 200,000 years ago. So in the preceding million plus years, humans existed together in one interbreeding population. That's what that long root looks like before the split. And then notice this nested branching pattern. Here's a bunch of mutations, and all of those mutations are common to all of populations studied today. Then another set of mutations separates one group of people studied from all other people worldwide. And this group of people exists in sub-Saharan Africa. They're called the San. Here's some examples. These are the San Bushmen of the Kalahari Desert. They're modern hunter-gatherers. And they are the most genetically diverse population of humans ever studied. Every other group in this light blue box is slightly less or much less genetically diverse than the San population. And then if we go down the line at all of these branches, we can see that there are sets of mutations that separate subsequent groupings. So this set of mutations separates the blue populations from all subsequent groupings. Everything in blue, the San, all the way down to the Yoruba, these are sub-Saharan African populations, and then we have everybody else. And these groupings continue to get more and more less and less genetically diverse. And I hope you get the point. At every branch pattern, there is a set of mutations represented by these blue stars that separate all the groups downstream of that branch from all the groups above it. This is a set of mutations that is shared by all of the descendant populations, but it was not inherited from an ancestral population before that split. So within this small ancestral population of humans or human ancestors that were interbreeding for about 2 million years, there was a lot of genetic variation that evolved, separating us from chimpanzees. And most humans inherited all of that. And then the branching started. So after the branching, well, maybe you could say that genetic races sort of exist, but there's a large number of sub-races and sub-sub-races, and none really correspond to today's racial categories. So like you could say, like a San race exists and then everybody else belongs to a non-San race. But that doesn't match the racial terminology used in the US, and it doesn't really help us, or today's racial terminology doesn't really help us understand the true pattern of human genetic variation. So what's up with that? This genetic evidence, the shape and the branching pattern of the human phylogeny, tells us a little more than what I've shared so far. Another way to look at this last 150,000 years of branching is to think about something called serial founder effects, where there was a recent African origin to all of humanity. Then there were successive population splits, one after another. People moved around, and local populations grew. Let me explain this further. Serial founder effects can be thought of like this nested children's blocks, where there's an original gene pool represented in the base, and that's the ancestral population. It has the most genetic diversity. And then each generation, some people move away from home while most people stay behind. And these migrators carry only some of the genetic diversity of the previous step with them, such that successive generations represent a whittling down of the genetic diversity found in that original gene pool. After serial founder effects, which population has the least genetic diversity? Can you point to it in this children's block diagram? It'd be this one, which represents the population furthest from the homeland of all humanity. So, under these expectations, understanding how serial founder effect works, when we look at how diverse our populations of modern humans, we can infer how long they've been living in different regions. And that's what this phylogeny tells us. It reveals the path and the timing of ancient migrations. We know that the most genetically diverse people live in Africa. And all subsequent steps out of Africa lose a little bit of genetic diversity. And so we know that these are ancient times. People were traveling on foot across the globe. And the populations today that have the least genetic diversity are those that are furthest through these paths of foot travel from our shared African motherland. And that would be the very tip of South America. And what we're seeing on the human phylogeny here is the most genetic diversity is within populations towards the top of that family tree, and the least genetic diversity down towards the bottom. And these represent, at the bottom, populations that are alive today who got to their current locations from a history of migration that took a long time out of Africa. Here's another way of looking at it. It's a nested Venn diagram. So if this is the genetic variation in humans, the most variation is represented by this big red circle, and that is the amount of genetic variation that exists in the gene pool among African populations. So Africa has the most genetic variation, and almost all non-African variation is also found in Africa, and that's why the circles are on top of each other. More recently colonized regions have a subset of the genetic variation that was found in the previous step along that ancient migratory route. But interestingly, some variation is specific to one group or another, and that's represented by these tiny non-overlapping bits of each green, blue, and purple circle. These region-specific bits of DNA exist because new mutations occurred in just one person within that region. And most new mutations are lost by chance due to genetic drift, or some persist by chance because they got lucky during genetic drift. But some are interesting because they increased in frequency due to natural selection if they were highly advantageous. And you know about some of these, like the lactase persistence allele and the sickle cell allele. They became more frequent in populations within these regions because they offered a survival or reproductive advantage. So what happens when a highly advantageous mutation occurs? So like sickle cell allele protects against malaria, lactase persistence alleles allow dairy digestion. Well, it turns out another example that you will learn about in the case study for this unit is melanin producing alleles, which determine a person's skin color. And to give away the ending of the case study, it turns out that the mix of melanin alleles in each person, they inherited them from ancestors. Let's see, we all inherited our melanin alleles from our ancestors, and our ancestors had evolved the melanin alleles that balanced the need for folate and vitamin D, these two essential vitamins. And so please read the case study if you're interested in that topic. It is fascinating. So basically, the scientists looked at people's skin pigmentation and compared it to the amount of UV light from the sun that reaches the Earth's surface. And here you can see that along the equator, as you would expect, that's where the most UV light reaches the Earth's surface. Well, melanin is a natural UV sunblock. And UV can be damaging. It can be damaging particularly to the developing neurological system, particularly of fetuses. And so how this works is that UV gets through a woman's skin and it degrades the folate in her bloodstream. So to prevent folate degradation, melanin evolved as a sunblock. However, UV light is also necessary for survival because it promotes the production of vitamin D. So if too much UV light is blocked, then people can't produce enough vitamin D to remain healthy. And so the blending of skin tones from the equator to the poles appears really well matched to the amount of UV light getting through the atmosphere in each latitude. So we have these latitudinal gradients of skin tone. And that's what the case study is about. There's another example of region-specific beneficial alleles that you read about in a previous case study, and that's the CCR5 delta 32 mutation that is known to protect against certain types of viral infections, including HIV. This mutation affects the receptor that HIV attaches to in order to invade host cells. So it changes the shape of the receptor such that HIV can't dock on that cell and cannot infect the cell. But this particular mutation is so prevalent. It's in around 10% of Europeans or people who trace part of their ancestry to Europe. And 10% is big. Remember from a previous lecture that polymorphisms are considered common if they're in 1% of people. So this one's in 10% of people. That's a degree of magnitude more frequent. And HIV is just too new. It dates only to the 1920s. And it originated, we know, in Africa. So why would Europeans have this mutation that protects against HIV? And if you recall from the case study, scientists think that it's because the same mutation originally evolved because it protected against a previous type of viral infection, one that plagued particularly Europe throughout the Middle Ages. So for several hundred years, it caused something called hemorrhagic fever, which was very deadly, way more deadly than coronavirus, even more deadly than Ebola. In some historical instances, it appeared that hemorrhagic fever killed every single person that came down with the illness, so a 100% fatality rate. That's really, really scary. The current coronavirus has a 3% fatality rate, and we're freaking out about that, and that's high too. But imagine if it was 100%. Tragic. And so you can imagine that would be really strong selective pressure. So over several generations of this particular hemorrhagic fever virus being around, the survivors who populated the next generation were more and more likely to have the CCR5 Delta 32 mutation that basically acted like a condom around their cells, preventing the virus from invading those cells. So if you haven't read that case study, please do so. It's pretty interesting. How common are these highly advantageous and region-specific alleles? One initial analysis studied the complete genomes of 179 people from all over the world, and they focused on sections of the DNA called SNPs, single nucleotide polymorphism. So these are 15 million variable regions of ASTs, Cs, and Gs that represent almost all of the variation seen among individuals in the human genome. And what this analysis found was that you really had to compare two populations against one another because there wasn't a single region-specific allele that only happened in one place to the exclusion of all other places. So here are some pairwise comparisons from different populations. Between European populations and various groups of Chinese and Japanese populations that were sampled, 2 differences were found. That means 2 letter differences, H's, T's, and G's, 2 out of 15 million. So we're talking really small numbers. If we compared Europeans versus Africans, 4 differences were found, 4 out of 15 million. And this includes the famous sickle cell allele, which we know spread into southern Europe after it first evolved in Africa. And 72 differences were found between the Chinese and Japanese populations that they lumped together for statistical reasons versus African populations that they lumped together. So we're talking really small numbers. 2 plus 4 plus 72, so 78 out of 15 million. So basically these region-specific alleles are super, super rare. These few differences are evidence against the idea of a biological or genetic concept of human race. In response to this, students often ask me, well, if race isn't genetic, then why are forensic scientists so good at identifying the race of people who maybe left blood behind at a crime scene, or the race of someone whose bones were found? So maybe they were a murder victim and the police are trying to get their remains back to their family. And I think these questions come from watching TV shows like CSI, but it is a valid question because this is done in real life too. So I wanted to take a minute to briefly explain how scientists do that. The first step is to make an assumption about the likely ancestry of the person whose bones or blood, so the remains that you found. And the scientist makes this assumption based on where in the world the remains were found. So like if the body was found in Tucson, the scientists might take a first guess that based on the demographics of Tucson, this person might have some mix of ancestry from both Europe and the Americas. And based on that assumption, they choose to evaluate variants that have a high frequency between these two ancestral populations. So you're not looking at the whole genome, you're just looking at the bits that are already known from previous studies published to be very, very different between European populations and Native American populations. Now, if the scientists guess incorrectly, then their analysis isn't going to turn up any results and they have to start over with a different guess. So even when one guess gives a initial, I don't know, the first guess doesn't fail, you still can't really narrow down like this person is like 40% European and 60% American. Even then, scientists have to consider additional traits. And so you might look at additional pieces of DNA or additional evidence from bone characteristics, a set of features that differs in frequency from place to place, even though no feature is known to occur in one location to the exclusion of a different location. So I hope this makes sense, that different sets of features are required for each pairwise racial comparison based on the scientist's initial guess. And when they guess wrong, they have to just start over. So it's a little bit of trial and error. Another question students often say is like, I can see differences between races, so what do you mean it's not real? It's not biologically real. So there is geographically patterned genetic variation that traces to ancestry, but it's not racial. And the classic example is skin color, which varies geographically based on how close your ancestors lived to the equator. But that's not a biological racial feature. It's common to all equatorial groups, so is there an equatorial race? Nearly the full range of human skin color exists in every region. And this is true of other supposedly racial features as well. They're also geographically distributed in relation to environmental conditions. And part of lab this week leads you through an exercise in which you're supposed to be convinced of this. The point is, you couldn't see races and recognize these differences between people until society taught you how. You learned probably from a very young age to cue in on certain physical characteristics between people and learn to identify racial identity based on a social construct. Nearly zero phenotypic traits occur within one population of people only. It blends from place to place. Therefore, phenotypic traits do not reliably align with racial categories because race is defined socially, not biologically. This social construct so profoundly influences our lives that we're going to spend an entire lecture thinking about that next week. And as one example, I shared with you in D2L a link to, well, it's an Under Armour ad, but Misty Copeland is in it. And Misty Copeland, if you don't know who this is, here's a picture of her. She is the first African American principal dancer of the American Ballet Theater. And if you know anything about ballet, you may have a preconceived notion that it's famous in Russia, it's famous in Europe, and ballet dancers historically are mostly white. And so you can hear Misty Copeland recount some of the things that she was told after being rejected from ballet auditions. And some of the things are, I mean, it's all veiled racism in these comments. And one of them that strikes me is the reviewers will say something about her body proportions, but she wasn't actually measured during her auditions. So if you want to be infuriated by racism, you can listen to her recount this experience as a ballerina. So back to today's question, are there biological races among living humans? If so, how different have we become? Are there innate biological differences between modern human groups? Nope. Because race is not genetic. It's not a biological construct. It's a social concept only, not a biological one. Next lecture, we're going to talk about how race predicts health independent of genetics, independent of socioeconomic status, independent of any sociocultural factors that people have figured out how to study. And so we're going to explore why race predicts health, even though it's not a biological construct. Spoiler alert, there is substantial evidence that racism plays a key role in our health, and this has been studied on many different levels, both individual racism and institutional racism. So that'll be next lecture, but before then, please complete Lab 5. There's 2 separate activities. One has to do with race, and you'll be directed to a website that has some interactive activities, so you'll need an updated Flash player. So for this activity, you'll examine the concept of race used in the United States, and it will challenge the notion that phenotypic traits reveal anything about your geographic ancestry, because ancestry is genetic while race is purely a social construct. The second lab activity for this unit I haven't talked about in lecture yet, so there will be significant D2L explanation you'll have to read. And what this activity requires for you to do is download a real-life data set from D2L that came from the US FBI. You're going to evaluate this data set to see who was likeliest to be arrested for different types of crimes, males or females. The point of this activity is to evaluate whether US crime statistics match sex typical behaviors that have been studied by behavioral ecologists in many different cultural contexts around the world. And because people who were born male versus people who were born female tend to typically, but not always, in all of these different cultural constructs behave in some stereotypical ways, that is considered evidence that there are biological differences in sex typical behaviors. Now, when I say sex, I specifically do not mean the social construct of gender. So male, female, these words refer to biological sex, contrasting the words man and woman, which reflect the social concept. Same with masculine, feminine. These are social concepts of gender. And so we're ignoring gender and we're looking at biological sex only. So read the D2L lessons to get more information about that, and I hope that you enjoy these lab activities. Bye.